If you open a book about the ocean, there's a good chance that the vast majority of the species shown to you in it. Whales, tuna, salmon, hammerhead sharks, jellyfish, turtles, octopuses, and sperm whales surprisingly make up only a small fraction of the ocean's total living mass. Oceans whose main inhabitants are unicellular organisms, which make up about 2/3 of the total biomass of marine organisms. I'll say it again, partly for you, but also a lot for myself. 2/3 of all ocean life by mass are invisible to the naked eye. This soup of microscopic life is largely made up of phytoplankton, unicellular organisms that, like plants, are able to use the energy of the sun to produce oxygen from carbon monoxide, and nutrients. And without the existence of these tiny organisms, absolutely none of the giants of the oceans that I listed earlier in the introduction could exist. For a long time, these invisible beings were largely forgotten among living things. Fortunately, oceanographic expeditions, like the recent one I joined on the schooner Tara, are helping change that. For nearly 2 weeks, they allow this fascinating microscopic world to be brought back to the center of the attention it deserves. These microscopic organisms are the main research focus of this oceanographic sailing ship, which has sailed the world's seas for 15 years, to immerse themselves in the study of living beings that are too small to put up lasting resistance to the movement of water masses carried by the currents, commonly called plankton. But before explaining to you how these missions have shaken up what we thought we knew about the diversity of living things, I need to show you just how much this microbiome really weighs in. Consider that the unicellular organisms making up phytoplankton are sometimes so numerous that they actually become visible across large portions of our ocean surfaces. So vast that you can only truly grasp their scale by taking a step back and looking down from above. Individually, phytoplankton are tiny, almost always invisible to the naked eye. But when these unicellular organisms bloom by the billions during events called blooms, they can form gatherings so vast that they become literally visible from space. The colored patches you're seeing on the screen right now, captured by observation satellites, are gigantic phytoplankton blooms that can spread over astonishing areas of tens or even hundreds of kilometers wide. These seasonal blooms mainly appear at high latitudes in late winter and early spring, along the equator in the Pacific and Atlantic Oceans, or on coasts and continental shelves, where rivers flow into the sea and where cold currents rise from the depths, rich in nutrients. Here, off the southwest coast of England and in the North Sea at the end of spring, it is common to see from space massive blooms of unicellular phytoplankton forming on the surface, where intense winds mix the shallow seawater and bring up nutrients under the returning sun. And since these phytoplankton blooms concentrate where powerful currents prevail, it is common, after strong storms, to see these colorful microscopic creatures. They draw the direction of water movements, the flow patterns, like tracing the outlines of whirlpools. The colors of these blooms reveal which species are proliferating there, because many of them have very specific pigments and reflect different types of light, which can appear green, red, or brown. And now the Black Sea turns white. Off Mauritania's coast, this green water gets its color from one source: a high concentration in the water of a type of phytoplankton called diatom, a microscopic algae with glass shells that encapsulate a lot of chlorophyll. In these other natural color images of the Baltic Sea, the vast greenish patches, some of which can reach hundreds of kilometers wide, are caused by blooms of unicellular organisms, very different from diatoms, but also performing photosynthesis, cyanobacteria that reproduce in this almost enclosed sea nearly every year at the end of summer. To grasp the huge size these organism patches can reach, consider that, in this image, the dark straight lines are all produced by the passage of ships which with their wake cut through the blooms. In 2019, cyanobacteria blooms have covered up to 200,000 square kilometers of the Baltic Sea's surface, nearly half the size of Sweden. If cyanobacteria blooms reach such proportions in this region of the world, it's because this sea, which is almost enclosed, is continuously supplied with phosphate, a nutrient coming from runoff. Rainwater irrigates the farmland abundantly supplied with fertilizer that surrounds the Baltic Sea. The rapid formation of so much life in just days may seem like great news. These immense blooms are indeed most of the time an opportunity for great feasts for all kinds of marine animals. However, these oases of life can eventually become layers of death. Large amounts of certain microalgae may turn toxic, and the vast phytoplankton created can, when dead, sink to the ocean floor and foster huge colonies of bacteria that decompose them, depleting dissolved oxygen in the water. so quickly that they end up suffocating? Sick organisms create dead zones nearly devoid of life, where bivalves and crustaceans die from lack of oxygen. Like many fish that, if they don't escape quickly, lose consciousness and die from asphyxiation. To better understand the seasonal activity and large-scale extent of these blooms, you have to dive into this animation created by Massachusetts Institute of Technology, which shows us the changes over time. Each color represents a dominant phytoplankton species on the ocean's surface. As life increases, it becomes more opaque. Cyanobacteria, dinoflagellates, haptophytes, or diatoms are all delightfully exotic forms of life that, just like terrestrial plants, live according to the rhythms of nature. The seasonal proliferation of diatoms, shown here in red, is almost solely responsible for the explosion that occurs every year in the spring in the high latitudes of the krill population, which will then feed whales, seals, leopard seals, squids, penguins, albatrosses, and a large number of fish, to name just a few. And so, even though they are tiny, these invisible algae that make up the phytoplankton together form the backbone of the ocean. That's it. By monitoring ocean surface activity and color, Kwon estimated that marine phytoplankton makes up half of the planet's primary production. By using the sun's energy, these photosynthetic microorganisms transform sunlight, carbon dioxide, and water into sugar and oxygen, or in other words, into nutrients and usable energy for living beings that can't eat sunlight. That's why photosynthetic organisms are called primary producers, meaning organisms that are self-sufficient and able to autonomously produce organic matter, on which the entire food pyramid will then be based. All crustaceans, invertebrate fish, deep-sea creatures, sharks, and whales rely on phytoplankton to survive. And yet, these unicellular organisms are not actually on their menu. They are only the main course for almost all zooplankton. Small crustaceans like copepods, shrimp, and krill also drift in the current. And after gorging themselves on phytoplankton, they will be eaten by small carnivores, fish, and cephalopods. Like starfish, which themselves will in turn in part be consumed by larger predators and so on, up to the last link in the food chain. If marine phytoplankton had shoulders, they'd carry nearly the whole marine ecosystem on them. And by extension, indirectly, 600 million humans who today depend on fishing and aquaculture, making a living, one way or another. If you've eaten a small slice of tuna recently, then you've very indirectly put more than a ton of seafood on your plate. 1 ton of phytoplankton can feed 100 kilo of zooplankton, which then feeds about 10 kilolice. Crustacean fry and small fish can feed 1 kilo of medium-sized fish, which then can feed 100 grams of tuna. And here's where it gets even stranger. It's estimated that in the ocean, there are 5 times more consumers than producers. Whereas on land, on farms, primary producers, plants, make up 80% of the total biomass. To really grasp how strange this is, imagine a savanna with 5 times more lions than wildebeests. Or if you prefer, a meadow packed full of goats. Still, all hoofed animals would eat only the grass beneath their feet. This kind of setup, where there are far more consumers than producers, doesn't exist on land, while it's the norm in the oceans. And if this feat is possible there, it's because photosynthetic unicellular organisms, in the presence of adequate nutrients and light, are able to renew themselves at incredible speed. Just think, unlike trees which with their slow pace can live and grow for decades or even centuries, unicellular organisms such as diatoms can replicate once a day while rarely living more than a week. And it's thanks to this abundance of life on a microscopic scale that the marine food web can entirely depend on it. A small population 5 times lighter than itself, a small people of the oceans who not only feed almost all the large organisms living in the water, but also contain forms of life whose diversity and extreme importance we only recently became aware of. Over the past 300 years as biology developed, we have mainly sought diversity in the great tree of life, where we thought we would find it, among plants and animals. And then, over the past few decades, with the introduction of new analysis techniques, first microscopy and then genetics, we began to discover the worlds of viruses, bacteria, and fungi. All the while, for a long time, we overlooked living organisms that are almost never mentioned, even though they should be making headlines everywhere, given their importance to life on the planet. Protists. A large part of phytoplankton belongs to this strange unicellular eukaryote type. That means their cells, like ours, contain most of their genetic material inside a nucleus. But protists are neither animals nor plants, and yet many of them are incredibly complex, and very often they perform photosynthesis and they eat. The analysis of samples collected during the expeditions led by TARA has revealed part of the unsuspected richness of these tiny forms of life. By tripling the estimated number of protist species and showing that there may be 10 times more eukaryote species in the ocean than previously thought. For example, it was discovered that a large part of the planktonic eukaryotes living in the sunlit upper layer of the ocean are diplonamids, a family of living beings you've most likely never heard of, even though they are probably by far the most diverse group of marine organisms. Today, this microbial life, visible in groups from space, forms a new jungle to explore. Studying life on our planet without considering it is like working a night shift with some radar screens off. You move forward in the dark, ignoring the diversity and scale of what surrounds you. For scientists, studying these microbial communities, which reproduce and evolve quickly while being very sensitive to disturbances in their environment, is a way to take the pulse of the oceans and assess their health. In this way, these microscopic organisms are like the canary in the coal mine, because they make it possible to raise the alarm as well as to better understand and anticipate the effects of climate change. It has thus been shown that with the increase in the temperature of the upper layers of the ocean, populations of diatoms, the most widespread phytoplanktonic algae on our planet, decreased by more than 1% per year between 1998 and 2012. Phytoplankton fills our plates, food for the mind and for research. It still has many secrets left to reveal during the ongoing Tara expedition. Focuses on how human activities affect biodiversity in European coastal ecosystems. The sailboat is just one part of a larger structure. Thus, using techniques developed by the European Molecular Biology Laboratory, we are now able to cryogenize samples of 1 milliliter of seawater without destroying the structure of its inhabitants. By exposing them to 2000 bars of pressure and temperatures below minus 100 degrees for a very short time, we can vitrify the water, turning it to ice without forming crystals. After that, we replace the vitrified water with a kind of resin before bringing the plankton sample back to room temperature and pressure. Which means, today, with this technique combined with very high-resolution microscopy, we are able to see the preserved structure, the delicacy of these organisms with unmatched precision and in 3 dimensions. Now that you have seen the invisible and know that nothing resembles each other less than 2 drops of water, I hope you will look at the ocean and its microscopic richness differently. A huge thank you to the entire Tara team for allowing me to spend 2 weeks aboard the sailboat. They helped me discover and appreciate the microscopic world. Doing science at sea can be as enjoyable as it is complex and physically demanding for both scientists and sailors. Because during my stay on the schooner, we had only one brief window of good weather, the rest of the expedition, the filming conditions looked more like this. And despite all the threatening clouds, the pitching and rolling, the moods on board remained invariably cheerful again. A huge thank you to them. A big thank you to Christian Sardet and the Macronauts for allowing me to share a part of it with you. Magnificent images they create of these myriads of tiny aquatic creatures. You can find them at www.planktonchronicles.org and in the book Plankton, At the Origins of Life. Even more of these striking photos.